So we're talking about chapter 7, so immune system. Okay, so we're looking at the adaptive defenses. We've been talking a little bit about the non-specific defenses, and now we're talking about the more adaptive, more specific types of defenses. So when you take a look at your adaptive defenses, what you're looking at is essentially a system that's designed to do recognition of whatever the foreign thing is that you are attacking, right? The bacteria or the virus or the molds or whatever happens to be on the agenda for your immune system to attack. And so what they respond to are basically what's called antigens. So basically an antigen is a molecule that provokes an immune response. So pretty much anything that doesn't look right. If it doesn't look like it should be part of your body, it goes after it. If they're abnormal membrane proteins, they will go after it, right? So even if they have your own self-tag, which a cancer cell would, they will go after the cancer cell, not on the basis of your self-tag, but on the basis of the fact that your cancer cells basically produce lots of abnormal proteins. So you can kind of imagine your immune system is kind of like being a big surveillance system. And so they're kind of constantly patrolling through your body, and they're looking for a couple of things. The first thing they're looking for is what's called your self-tag, right? So there's a glycoprotein group called your major histocompatibility complex that it basically represents like your self-tag. So every single cell in your body has this tag on it. And so it's kind of what defines my cells as mine. And my immune system, it grows up and is trained to recognize my cells. It reads my name tag. Now, if I were to get a liver transplant from Bob, then Bob's liver cells would read Bob. It would have Bob's name tag on. And my immune system would recognize that I am not Bob and Bob does not belong in my body. It would go on the attack. That's the basis of tissue rejection. And so looking for those, and obviously anything that's foreign to your body is not going to have your self-tag to it. And so basically, your immune system is just purely identifying, do you belong here or not? This is Mark's body. You better have a Mark tag on you, right? If you don't, then I'm going to kill you. That's basically how the immune system works. The other thing it looks for is even if my cells do have Mark tags on them, there's another group of cells in your immune system whose job it is to kind of patrol around and to sort of look for suspicious behavior, if you will, right? This is kind of the idea of this abnormality search. So for instance, a cancer cell in my body would have my self-tags on it. They would say Mark on them. And so they would escape that first level of surveillance. However, typically speaking, cancer cells are kind of rogue cells. They kind of do what they want. And the proteins that they make are not normal to the type of cell that they are. And so your immune system can screen for the types of proteins that you're making. And if it looks like it doesn't match up, then it can kind of basically say, wait a minute, this is not right. You're not supposed to be making this type of protein, so I'm going to have to kill you. Think of it this way. I mean, if you've got a baby bottle factory, it should be making baby bottle components. You screen that factory and you realize it's making bullets and grenades. Something doesn't add up there, right? That's an abnormal use of that factory. And then at that point, then what you do is you demolish that factory because clearly that factory's gone rogue. And whoever owns that factory is making weapons, not baby bottles. Right? That's kind of the way your immune system sort of works. So your adaptive defenses have two major groups of cells that they depend on. The first one are the B cell lymphocytes and the T cell lymphocytes. Now, B and T cells have specific receptors for specific antigens. So they're looking for specific problem molecules that would be an issue. And so these are basically membrane proteins that will be designed to bind or search out particular antigens. So for instance, they'll kind of be a cell and they'll look like they have a little protein that kind of has this little receptor embedded in them. And so they're looking for very particular types of proteins that they're designed to recognize. And when they recognize it, then they can get activated, right? So basically they recognize that they found the bad guy. Once they found the bad guy, then they can go on the attack and they can call it their friends, right? So generally speaking, each of your lymphocytes has one type of receptor. And then when the receptor meets its antigen, it'll fit together, kind of like square peg, square hole. And then that's what will be the basis of activation of that particular lymphocyte. So there's two major pathways of adaptive immunity that you're going to see are activated by these lymphocytes. One is what's called cell-mediated. So basically, it's like cell-to-cell combat. So if you want to think about it in terms of like the war analogy, cell-cell combat is kind of like hand-to-hand combat, right? Like kind of like we used to do back in the old days before we invented guns and things like that, where warriors would have to charge into battle and they would have to like meet each other face to face with their swords drawn and that sort of thing. And then you would have to kind of do battle hand to hand and person to person. And that's kind of what cell mediated is like. Antibody mediated is basically kind of more like artillery. So if you've ever seen an artillery unit, basically they've got the mortar launchers. There are usually those like gigantic guns in there. You put the mortar in there, you basically fire it and it shoots the mortar across a large distance to a distant enemy that you aren't anywhere close to. And the idea is you drop a mortar shell in the middle of them and you blow them up, right? The idea is if you can scatter the enemy, kill them, or scatter them, you'll win the war, because if they're running from mortar fire, then they're not going to have a lot of time to come and hurt you or kill you. And so that's kind of more like long range combat. That's essentially what the antibody mediated strategy is like. So the T cells are the ones that are basically driving the cell mediated immunity. And so basically what they're looking for is they're looking for other cells that are presenting a specific foreign antigen. These are called antigen presenting cells. Or APCs. And so this is part of one of the duties of some of your cells that you have is to present antigens so that your cell-mediated immunity can find them and then kill them before they start killing you and harming your tissues. The antibody-mediated immunity is basically what we refer to as the humoral immunity. This is the one that is making antibodies. So you're making antibodies that will basically flood your system, and then they will go and seek and bind to antigens in your body, wherever they will find them, and they immobilize them, and then they destroy them. And so this is kind of what it looks like from kind of a flow chart. So here you can see you have your adaptive immunity at the very top, going to split into your cell-mediated and your antibody-mediated. Now, generally speaking, your cell-mediated has got to have presentation of an antigen, which is a little bit different than your B cell or your antibody mediated. But ultimately, this antigen presenting cell is going to present this abnormal antigen to your cells, and that is going to basically tell your T cells, this is what the abnormal guy looks like. And so what's going to happen is typically you'll make two different types of cells in this cell-mediated situation. So the cells that you have are what's called cytotoxic T cells. These are the warriors. These are the ones that will go after the pathogens, the cytotoxic T cells. but they also will make what's called helper T cells. So these guys are the regulators. These guys will regulate not only the T cell response, which eventually will basically regulate the cytotoxic T cells, who are the ones who will basically go after the pathogens, and then they will either gobble them up through phagocytosis, or they will basically create apoptosis in those infected cells, like if it's a cancer cell or something like that. So this is cell suicide is what this is. Apoptosis. So basically the cell kills itself. It will also regulate the activities of the B cell lineage, which are the antibody producers. So in the case of the B cells, the antigen itself is what's going to stimulate the B cell or activate the B cell. So the B cells have a particular receptor that will bind to a specific antigen. And when it binds a specific antigen, it activates it. And so you'll make lots and lots of B cells. You make a couple of different types of B cells. The first one that you're going to be making is what's called the plasma cell. The plasma cell are the cells that are responsible for making the antibody. So what you have on the surface of your B cell is effectively the answer to your problem. I've located this antigen, I've recognized it, and now this holds the information necessary to duplicate that, right? So once you've solved the problem, then you duplicate the solution so that everybody has a solution to the problem. And that's what's happening with the plasma cells. The plasma cells will basically produce lots of antibody. And then the antibody will basically flood your system. It'll go throughout your plasma, and it stays in your plasma. And as it runs into antigens, bad guys, it'll grab onto them and immobilize them and destroy them. Okay. If they're part of a cell, they will basically destroy the cell, like a bacteria or something like that, right? And so that's basically what the plasma cells will do. Now, this is basically nothing really different, if you think about it, from what we saw with the non-specific piece, right? Because so far we haven't, I mean, yeah, we've recognized stuff, but we haven't really solved a lot of our problems yet, have we? Because what happens if we get infected by this guy again? We have to go through this whole recognition rigmarole all over again, like we're starting over, right? Now, remember, the thing about the adaptive system is it doesn't just recognize, it also remembers. So the adaptive recognizes and remembers. So how does it remember? It remembers because when you find the correct match of either cytotoxic T cell or the B cell, what they do is, in addition to creating cytotoxic T cells, they also create what's called memory cells, T cells and B cells, that will basically form a profile or a memory of that attack. This is the reason why when you get infected again, it's not as bad, because your body already knows the enemy. And so rather than going through that strong learning curve, right, where it's kind of a very slow learn of your enemy, you already know your enemy now. You know your enemy well because you've got a memory file on your enemy. So as soon as your enemy tries to come in again, boom, you can just jump straight to the attack and you can eliminate them. This is one of the reasons why whenever you acquire immunity to something, like say, for instance, a particular strain of flu, you may get sick with the flu. And basically the reason why you're getting sick is because your immune system is trying to identify the enemy and is trying to learn how to recognize that enemy. And while it's learning, the enemy is making damage on your body. That's where the symptoms are coming from. And then once your immune system figures it out, then that's when the symptoms start to abate and your immune system starts to get the upper hand. And then eventually your immune system wipes it out. Now, what happens when you get infected by the same strain of flu a second time? Well, your immune system already knows that flu strain. So there's no learning curve this time. So when that flu enters in and tries to do damage to you, can't get anywhere because the immune system says, oh, I know you. I beat you earlier in the flu season. Guess what? I know how to beat you now. So you're not going to be able to get the upper hand on me while I try to figure you out. I've already figured you out. So I can just jump straight to making antibodies and killing you and attacking you. So the flu never gets a foothold so that it can start creating symptoms. That's basically how immunity is supposed to work. The adaptive immunity. So you retain a memory of the attacker. So on the B cells, these are the antibody-mediated side of things. You have B cell receptors or BCRs. These are basically the protein receptors that are on the cells themselves. So this is where you bind to the antigen. So the antigen that's in your body will bind to only one type of B cell. You have lots of different B cells. One thing you need to understand is these receptors, these B cell receptors, are incredibly variable. Very much like a random number generator, right? So what you're doing is you're sort of sorting through this pile of B cell receptors looking for the match, right? It's like a needle in a haystack sort of a thing. That's part of what creates that learning curve, is it takes time to find that match. But basically, there's so much variation in these B cell receptors that you've got a match somewhere. It's just a matter of finding it, okay? And so once you find it, then what's going to happen is it's going to produce copies of itself. And then basically, it'll create multiple B cells of the same type. This is called cloning, right? So multiple clones, lots of the same type. This is kind of what it looks like. So here's all your B cells. Notice you got all kinds of different B cells for different types of antigens. But the antigen that you're dealing with is not going to match to all of them, right? Think of it this way. It's like having one of those jailer key rings full of keys, and you've got an unknown lock. You know your key is in there somewhere, but you've got this massive key ring full of keys. So what do you start doing? You start trying the keys, right? Nope, not that one, 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 and so on and so forth until, there it is. Now, once you found that key, in this case, to a place that you really want to go, what do? You separate that key, you make copies to everybody else who needs access to that room. That way you have more efficient. And when you need access again to that room, you don't have to go through your jailer's ring, do you? You have the key now. You know where it is. Typically, if you're like me, you'll like put some paint on it or fingernail polish or something like that, or some sort of a tag on that one. So you know that orange paint is front door, right? Green paint is garage door. And so that way you can identify and recognize the correct fit for the correct problem. Same thing's happening here. You got multiple solutions to problems you're not solving. The question is, where's your solution? So what happens is you basically screen through all these B cells. It takes a little while. But once you actually lock onto that and you find your correct match, your receptor that matches your antigen, then that's going to activate that B cell population. And that's going to start going through division, cellular division, and it's going to start making multiple clones. These are basically many of the same B cell. That's what clonal expansion is. And then you're going to create two different piles. The first one are the memory cells. This basically stores a profile of the antigen against later infection. And then you have the plasma cells, which are basically the ones who are going to be doing the battle for this round, right? So these are the fighters for this round. It's kind of like the military, right? So the military, you've got two different branches of tactical, right? If you take a look at the tactical branches of military, there's two different major branches. Number one is the tacticians or the tactical officers who are planning the battle for right now. We need to win the battle today. This is the battle we need to win, right? So they're focused on today's fight. There's also another part of the tactical that's responsible for collecting information and intelligence on the enemy, both in the past and also intelligence for the future. Because oftentimes when you go through a battle, the kind of, I call it the research and development side of tactical, they're collecting information of the results and the strategies of the battle that we just did yesterday. How did we do? Did we win? Where's our weak spots, right? What's going to be something, where are the strong points of our enemy? Where are the weak spots of our enemy, right? And then how do we parlay that analysis of our enemy and also of us into a winning strategy for today's battle? And then today's battle will inform tomorrow's battle. So we keep this sort of running memory and strategy associated with our battle, and that's what the memory cells are doing. So the plasma cells are going to be creating a ton of antibodies, which will go throughout the body and bind to any free antigen that happens to be out there. This is really helpful for like bacteria that produce toxins. And so you have to produce antibodies against those toxins to keep them from doing damage to your tissues. It's also helpful for situations where you have cells that will bind to antigens that are actually part of the actual bacteria itself, and they'll basically grab that bacteria. Now, once your infection is over and you no longer need it, then you can basically tell the plasma cells to go through apoptosis, basically cell suicide, so that you can do away with them, recycle their parts and things like that. But even though you don't have an active infection going on right now, that you still have the memory cells, right? So even though you send the soldiers home after the war, right, after they're decommissioned, they serve their tour of duty, it's time for them to go home and to live their lives. That's kind of what this piece is like this. So basically you're going home from war. Go home and live your life, right? Which oftentimes, if you're a soldier in war, is easier said than done. Just ask any veteran. So that's your go home piece, but it doesn't mean that you forgot how to win that war, right? Because all the tactical officers still are the keepers of that knowledge. They're the keepers of that memory. I guarantee you that if you were to get into the deep inner circles of a tactical strategy plan at the military level, there's going to be a lot of assessment, strength and weakness assessment based on what we know about the enemy from the past. But I guarantee you there's always going to be a branch of those tactical officers where they're looking back and they're thinking about previous generations of warriors, of like, D-Day, strategies that we deployed during D-Day or during the European fight during World War II, or the Pacific fight during World War II. And we'll be looking at those strategies and asking ourselves, why were some of these battles successful or why were they not? And can we duplicate that success today? The Romans were masters at doing that. So they would take a strategy that served them well in a previous battle, and they were the kings of being able to deploy that strategy in brutal efficiency to later battles. It made them very, very difficult to beat. The Romans were a very impressive army in terms of discipline and strategy and tactics and technique. Most other armies were mostly just a bit thuggish in comparison. They were just brutal and scary, but they weren't really smart about the way they fought. The Romans were very clever about the way they fought, in addition to being brutal. Not that they weren't, right? Because they were quite brutal. It's great to be a Roman, apparently, from what I understand. Not so great to be on the opposite side of the Romans, right? Not so great to be a Roman enemy. So let's take a look at some of the characteristics of the B cells. So obviously these guys, we mentioned this a few times already, these guys are going to be the ones responsible for the antibody-mediated immunity. They're going to be basically attacking the individual pathogens. These guys are matured. They're produced and matured. There's two different things going on here. So they're born in the bone marrow. They're matured in the bone marrow. That's the reason why they're called B cells. B stands for the bone, right? The bone cells. And so their job is to directly recognize the antigen, and then they go through that clonal selection, which then expands to create your antibody-producing plasma cells plus your memory B cells. Now, during the clonal expansion, oftentimes what will happen is that these B cells will also respond to cytokines from T cells, where the cytokines are signaling molecules. And you're calling for backup. Looking at the immune system, it's really, really helpful to think of it as a military type of operation, because it very much is, including military communications, right? Because when you have different types of responses from different divisions, or even from different branches of the military, Navy to Air Force and things like that, the communication between them has got to be very, very good, because the Navy has to understand what the Air Force is doing. The Air Force has to understand what the Navy is doing. Somebody's got to pull them all together, because if they don't, then the infantry, which is typically Army, on the ground isn't going to know whether or not they have cover from the sea, like for instance, what they thought they had with Normandy, or if they've got air cover, which they also thought they had at Normandy. As it turns out, the airborne did not really quite, they were great, but they did not quite do what they wanted to do. And they were further inland than Normandy, so they didn't really take out those machine gun nests. And the bombardment of the sea coverage was ineffective. And so basically, those soldiers who were hitting the beach were sitting ducks, because it was supposed to be that those machine gun nests were supposed to be taken out by coastal fire, right? But that requires communication across the entire branches, right? All the branches. And so the communication has to happen here too, right? So here you have a communication from one branch, which is the cell mediated, to another branch, which is the antibody mediated. And that's what the cytokines are doing. So the cytokines are talking back and forth to each other. And just like with D-Day, D-Day is a really good example, because it was multi-branch operation. There was a lot of individuals involved. It wasn't just Navy, it wasn't just Army, it was pretty much everybody was involved there. And also multi-allied, but there was one person in charge. And that person was, the history buffs. You guys got to pass your history class? Who was in charge of D-Day? Later became president of the United States. You guys got to watch. I was going to say watch Saving Private Ryan, but that shows you what that day was like. But it was Eisenhower, Dwight David Eisenhower. So basically, he was, before he was president in '52, he was the military officer, the commanding officer over all of the Allies. So he was coordinating with the British and everybody else that was working with us, most of the British. But he was the one in charge of all that. So he was coordinating, I think himself, I think he was an Army guy himself, but he was coordinating like the Navy, the Army, the Air Force. He was coordinating everybody, making sure everybody was doing what they needed to do, which was one of the reasons why, not only that because he was very popular as well, but that was also one of the reasons why people had no problem electing him as president, because to pull all the NATO forces, what would later become the NATO coalition, together in one battle and coordinate it, takes a little bit of administrative capacity that you want in a president. And so that's a coordinator. So who's our central coordinator here? It's another T cell type, but it's a helper T cells. So the helper T cells basically are the regulators of both B and T cell response. So they're kind of regulating all of it, just like Eisenhower was the regulator of D-Day, right? So all that information was coming through Eisenhower, and him and a very small group of people were basically making sure that everything was ready to go and was exactly as they needed it to go. So this also is one that's regulating the B cells as well, right? So who's telling the B cells to clone? It's the helper T cells. So these guys are directly responsible for the B cell cloning, making them basically produce more and more cells so that the majority of them will become plasma cells and basically make large amounts of body, kind of like your artillery, right? So these are basically, you're creating a lot of artillery cannons, and you're making a lot of artillery shells so that you can fight an artillery war, okay? And then, of course, some of them will become memory cells. You don't need a lot, right? but you do need some in order to maintain a memory, because you can always basically reclone these memory cells, re-expand these memory cells, and then fight the same enemy. And this basically, once you have your antibodies, which don't go away, by the way, and you have your memory B cells, your memory cells, then this basically will give you immunity to that antigen. So when you're asking whether or not you have immunity to a particular pathogen, you're looking for those two things: memory cells and antibodies. Okay. So once you have won the battle over your infection, then your plasma cells aren't needed anymore, and they go undergo apoptosis, which is basically cell death. We don't need you, and so they get destroyed, but you maintain that memory in those memory B cells, and so you're able to keep that for later use. Just like I can guarantee you, at places like West Point and Annapolis, and probably also at the Air Force Academy, you have the future generation of tactical officers. Probably there's an entire unit of studying what went into that day, the planning, the military strategy, how Eisenhower set that all up, and what made it successful, and what are some of the lessons. So I can guarantee you our future officers and future generals are at those academies learning the lessons that we learned from D-Day. So they're being trained. That's kind of part of the memory piece. We will maintain that memory of that battle for future use. So when you take a look at antibodies themselves, they're kind of a Y-shaped protein. They have two different arms, and they have two different main regions, heavy and light chain. They also have what's called a constant and a variable region. So the heavy chain basically is just a long polypeptide chain. That's why they call it heavy. And then the shorter polypeptide chain is called the light chain. And so associated with these heavy and light chains, you have what's called a constant region. Basically, there's no change here. It's always the same. Versus a variable region. That constantly changes. And so typically we'll oftentimes classify our antibodies based on this constant region, this non-changing region. Now you may be wondering, why do we have the variable region? The variable region, which is the V piece here that you see here. So this variable region is what changes. To match the antigen. Now, it's not changing in and of itself, right? Each B cell has a particular type of variable region. So when we take a look at the previous cartoon where we had the different B cell receptors, one was kind of more triangular shaped, one was kind of more square shaped, the other one was round shaped. So the different shapes for those antigens is based on the variable region. So each B cell receptor comes out a different shape. It's kind of like a key, right? Each one of these receptors has a different cut in the teeth, so to speak, of the key. And you can produce as many shapes of those key teeth as you wish, right? It's kind of like, this is the random number generator. If you've ever seen a random number generator, it's basically like lottery balls dropping out, right? How many different lottery numbers can you produce by just dropping that ball out of the ball drop machine when you do the lottery? You can generate a lot of numbers, can't you? Each one of them will be different. The chances of you getting the same number twice is pretty remote, especially if you've got a long sequence of numbers, right? The longer your sequence, the less likely it is that you're going to get both hits. That's exactly what these guys are like. These guys right here, the genes that encode these guys, will go through a process called recombination. Or what's called highly recombinant. So recombination is basically how a gene will shuffle its information and constantly mix it up. It's kind of like shuffling a deck of cards. And so some genes will do it a little bit, and some genes will do it a lot. These genes does it a ton. So they're constantly shuffling. So the chances of you getting the same shuffle pattern twice is pretty remote. This gives you a maximum number of these variable regions. So you almost have, I don't want to say quite an infinite selection of receptors to pick just about any antigen you want, but pretty close, right? It's served us well for the most part. It's been clearly enough to save us from most of the insults on our immune system. But that's what these guys are doing. So this is basically where you have these constantly shuffling polypeptides that are changing and sort of hoping to fit. Now, if they don't fit your antigen, that's fine. Guess what? You know what they do fit? They fit some other antigen that you haven't seen yet. It's just like a key, right? A key fits a lock. It may not fit your lock, but it fits a lock. There's a lock somewhere that key fits to. You just haven't come across it yet. So what do? You hang on to that key for safekeeping, because someday you may come across that lock. And when you do, you want to have that key, right? That's exactly what these variable regions are like. Okay? You just produce a ton of these different types of receptors, and then you sort through them to try to match them to your antigen. So those guys are going to be a component of both the large chain. You can see the heavy chain right here, which has a constant region in dark, tipped by the variable region, and then the light chain, which is right next to it. Again, you have a constant region in the tip and a variable region that's right next to the other variable region. So this is basically what an antibody looks like. So the variable regions are the ones that can basically match to your antibody. And so that's just a blab slide for what we mentioned before, that whole lock and key idea. Okay. So we take a look at the structure. We already mentioned that they're kind of Y-shaped molecules. Typically, we'll refer to these guys as monomers. So if you just have like one Y shape, that's a monomer. Sometimes they can be paired together as a dimer. So for instance, you can actually have antibodies that are connected kind of end to end. So that would be like a dimer. So that's one possibility, one strategy. Sometimes they can form clusters. So for instance, you can have these clusters of antibodies that kind of form these little star-like pentameters. We have 5 of them all kind of together like that. So they can form different types of shapes. So they can be brutally efficient at binding to antigens. So what happens when they actually bind to the antigen when they find it? Well, when they bind to it, they basically go through a process called neutralization. So essentially the antibody will coat the virus, toxin, or whatever. And so when it covers them, they've basically been neutralized, right? And so basically you've removed the threat. That sounds very military, doesn't it? The target has been neutralized, right? You can just hear them. That basically means that you've got it taken care of. It's no longer a threat. Right? Now, usually when you hear somebody say that in the military, it means that they've shot them, is basically what they mean. Right? But the idea here is this is kind of like the molecular equivalent of taking out the enemy, right? And so ultimately then, when they surround these antigens, what they create is this sort of what's called the immune complex. It basically clumps all of these antigens up because these antigens are all kind of bound up by the antibodies, and they create these big complexes of antibodies bound up to their antigens. And this basically attracts the white blood cells, which basically come in and phagocytize the whole mess. And by doing that, they basically eliminate all that debris and everything like that. Okay. So they'll come in and clean it up. So there's different types of antibodies, different classes of antibodies. I'm just going to touch on them briefly, because I mean, we can get into them deeply, but I don't necessarily know how useful that would be. Just some broad brush strokes, right? I mean, after all, it's not an immunology class. We want to make sure we don't get too crazy here, right? So we basically have them, we have 5 different types of what's called circulating antibodies. These are the things that are in the blood plasma. So you have IgG. So basically what the Ig stands for is immunoglobulin. And so we have IgG, M, A, D, and E. IgG, this is going to be the major one. This is the one we think about when we think about antibodies in our bloodstream. And so this is going to be what we see in our blood. We do have a little bit of that found in our lymph system and in our interstitial fluids in our tissues, but it's going to be mostly in our blood. And so its job is to basically seek out and bind to either pathogens or to toxins that the pathogens will create. So a lot of bacteria can themselves be pathogenic, but a lot of times what the bacteria will do is they'll secrete toxins that basically are noxious to our tissues. And so we actually form a symptomology that's associated with the toxins, right? And so a lot of these will actually come in and bind to the toxins. They'll locate and recognize and bind to those toxins. These are also the ones that will cross the placenta from the mom. So basically the fetus's immunity comes from mom. So mom's IgGs will actually, she'll donate her IgGs to the baby, giving the baby immune cover, right? While its own immune system is busy trying to be trained and built up. And so this is one of the reasons why a lot of times, we don't think about babies getting sick because mom's immune system is covering for the baby. Matter of fact, the way mom's body works is generally if some sort of a negative insult happens to the mother's body, typically what will happen is the mother's body will take on the brunt of the insult, shielding the baby. So it's not uncommon for the mom's body to sort of guard the baby very, very strongly, and will take on most of that insult, protecting the fetus. Okay, and that's true for a lot of mammals as well. This is also the one we typically associate with temporary immunity. So this is also the placental immunity is also temporary as well. This is just a table for you table people who like to study off of tables. It just kind of puts them all in one place for you. And that's just there for you guys. Okay, so kind of organizes them in one place. So what's on here is basically the list that we're going through. Okay, so that's just what that is. So the IgMs, these guys are pentameters. So those are like star-like antibodies. These are oftentimes the very first ones that a newborn will produce. So when the newborn's immune system is kind of starting igniting and sort of getting going, it'll start to make a bunch of IgMs. This is oftentimes also the first on the scene. This is the first one to appear in the blood after an infection will begin. So in the initial infection. And it's also the first one to disappear, the first one to go away once the infection is over. So you can think of this one as sort of like a first responder. So at the scene of an accident, who's the first one on the scene? Fire truck, right? But they're only there long enough to stabilize the situation. As soon as the ambulance is there and the police are there, guess who leaves first? Fire truck, right? They go back to the station because they have to get ready for the next first response call. And so these guys, the fire truck is sort of like the IgMs of the antibody world. These are also the guys who will activate that complement system. Remember the complement system, the one that basically popped the bacteria like a water balloon, basically putting holes in them so that they essentially popped. These guys are the ones also controlling a lot of the inflammatory response with the complement system. IgAs typically are coming across as either monomers or dimers. And so they'll have a Y-shaped type of antibody. Generally speaking, these are the ones that are kind of standing guard at major entry points in your body. For instance, in your saliva or your tears, your mucus, your breast milk, these are all areas where you can get entrance of outside things, right? So your saliva, obviously, because you're putting things from the outside world into your mouth. And so you typically have IgAs there for any kind of pathogenic entrance. So if you just basically ate a bunch of bacteria, your IgAs start to go to work. Your tears, right? If you get any kind of bacterial intrusion into your eyes, your tears basically have IgAs in them. They'll start doing battle with these bacteria. Same thing with your mucus. Your saliva doesn't just have IgAs in it, but it also has mucus in it as well. All your other mucous membranes, say for instance your nasal cavity, right, and other areas like that, they also have IgAs in it. And breast milk, right, because the baby is taking nutrients from the mother, but also the baby is also taking antibodies from the mother's breast milk as well. And IgAs are a really nice addition to what the baby is already making, because remember we said the baby's already making IgMs, and now mom gives the baby IgAs in addition to that to help kind of further bolster the baby's early immunity. And so the idea here is if we can do battle with the pathogens before they even get anywhere close to the bloodstream, that's the best. That's the best situation, right? We don't want the pathogen to get into the bloodstream. We want to get rid of them before they get to close to the bloodstream. That's what it is. It's a preemptive attack, right? So it's basically eliminating the conflict before a conflict is able to actually mount. So the IgDs, these guys are essentially a B cell related antigen, or excuse me, a B cell related antibody. So these are going to be actually on the immature B cells in the bone marrow, and they're part of the signaling system. I don't want to get too much into IgDs because you have to get deeper into the whole production pathway of B cells and things like that to kind of understand their role at a deeper level. And then IgEs typically are focused on worm infections, parasitic worm infections. They can also be increased under allergic responses as well. If you have bad allergies, you probably have high levels of IgE. Or if you have a parasitic infection, which hopefully you know, you can also have high levels of IgE as well. So you have a whole spectrum of antibodies that are basically at your deploy when you're talking about doing battle with all these different things. So you're not just talking about one particular type of mortar shell in this case. You're talking about a whole spectrum of mortar shells. Think of it as sort of like the guns on a naval ship. If you've ever looked closely at the guns on a naval ship, they're not just one type of gun. There's multiple different types of guns. Some are small and long range, but they do very little damage. Others are bigger and very damaging, but they don't have as much range. So if you're in close, you know, you have to use the big gun. So you have these different types of weaponry to help in whatever situation or scenario you happen to find yourself in. So monoclonal antibodies basically are situations where you have antibodies that are identical. They're going to be produced by plasma cells, and they're derived from the same B cell. That's what they mean by the same clone, monoclonal. The same clone of B cells. Generally speaking, we're talking about monoclonal antibodies. We're talking about lab-produced antibodies. And so we usually use these for things like medical tests and treatments and research and things like that. So we generate an antibody from a single clone. Now, this is compared usually to what we refer to as a polyclonal, which means you can create antibodies of the same type from different clones, right, of B cells. So this is kind of what it looks like. So in this particular case, the way we make a monoclonal is we take a plasma cell of a clone that we've identified with a particular antigen. And then what we do is we take a cancerous cell, it sounds a little odd. In this case, it's a myeloma cell. And cancerous cells oftentimes will divide without regulation. And they'll produce lots of protein, like too much. So it's not producing just enough protein for the cell, it's producing tons of it, like overproducing the protein, which is what we want, right? Because we want high production. And so that's the reason why we pick a cancer cell. And what we do is we fuse these guys together. So the idea being, maybe we can create a hybrid cell that has the overproduction of a myeloma cancer cell with the identity of the antibody production of the plasma cell. So essentially creating a high overproducing factory for these plasma, for these antibodies. And that's basically what we refer to as a hybridoma. So a cell that overproduces the antibody that we want. And this hybridoma can then divide into similar cells like itself, and it can basically produce tons and tons and tons of antibody. Oftentimes, this is important because if you ever have a vaccination where you get a vaccination of antibodies, right, then probably those antibodies were retrieved by some process like this. They were able to identify a clone of some sort with a helpful and therapeutic antibody, and then they were able to create a hybridoma and have that produce tons and tons of antibody so that they can put it into a serum and then inject those antibodies into you. Okay. So that's probably some sort of a version of this, not exactly the same perhaps, but some sort of an idea of what we see here. So that's a lot to say about B cell receptors. Now, what about T cell receptors? T cell receptors typically are unique receptors, and they're usually on the surface of the T cell. So they're different. What we say, they're different from the B cell receptors. So they're not the same. So the B cells have unique receptors that are B cell only. T cells have a unique receptor that are T cell only. The difference here is that T cells cannot recognize the antigen on their own. They need help. So that's where you get your antigen presenting cell. This is basically a cell that will display the antigen to the T cell and tell the T cell, look, this guy right here is the bad guy, go get him. Okay. And generally speaking, it's the macrophage. Why? Because the macrophages are often in your tissues phagocytizing the antigen. And then one of the things that the macrophages do is when they gobble up the bacteria, where the bacteria has proteins that are very unique and individual to the bacteria, right? So they don't occur in our body. So they would pass the abnormal test for the immune system, right? So what the macrophage does basically is it chops up the bacteria, and then rather than like using all those amino acids and things like that, what it does, it basically chops it up into bits. But then what it does something clever. It bundles it with the self tag so that the immune system can read a receptor, says, oh, okay, I see this is a self tag with an abnormal protein on it. So what it does, it displays that abnormal protein to the outside, hoping that the immune system cells, like the T cells, for instance, will see that. They'll read it. They'll be like, oh, okay, I see the self tag for a macrophage that has an abnormal protein. So this is the thing that I need to discover and to attack. Think of it sort of like an enemy, right? And kind of what you do is a kind of a warrior. You meet the enemy and you kill them. That's what the macrophages do, right? And then instead of just like, cremating them or whatever, what you do is you actually like cut off the enemy's head, and then you take it back to your camp and you show them the head and you say, listen, this is what the enemy looks like. If you see this or anything that looks like this guy, you got to kill him because this is what the enemy looks like. That's like an antigen presenting sort of a thing. That's sort of what the macrophage is doing. Now the army knows what to look for, right? As they're sort of going around, they kind of get an idea of what the enemy looks like. And so that's basically what these macrophages are doing. So they'll take these pathogenic pieces apart with the lysosome, and then they'll bundle it with their self tag, which is a major histocompatibility complex, right? This is your self tag. And you have two different types or two different classes of your MHC. You have MHC1 and MHC2. And so ultimately what's going to happen is you're going to bundle it with your MHC. Now, one of those MHCs you use for antigen presenting, and the other MHC you use for your self tag, right? So you basically say, listen, this is not antigen presenting. I'm not giving you the head of the enemy here. This is just me, right? So this is my self tag, my little name tag right here. So that's what one of them is. The other one is for presenting of the antigen. This is the enemy. Here it is, right? So that's what those two are for. Now, in humans, we typically will focus on the MHC2 classes. These are what's referred to as the human leukocyte antigens, or the HLAs. This is on all body cells because these are your self tags. And we have three major groups of the HLAs. First of all, it's A, B, and DR. And so this has different types of protein variations associated with it. There's different variants of it. And each person will have a specific combination of these variants, right? So it's like if I shuffle a card deck and I deal you a hand, each one of you is going to have a specific combination of cards. That is right? On that combination of cards that you have, whatever that hand is, that is like your identity. That's like your serial code, right? And so that becomes your basically how your self tag works. It's basically a serial number or like a barcode of these variants, these HLA variants that basically is all yours. And so you have a unique combination of these HLAs, except for, of course, twins who are genetically identical, and they will have the same because these are genetic in nature. So then once you have antigen presentation, then you have to move on to clonal expansion. And so typically when you get to clonal expansion, you're going to start making many copies of your activated T cell, just like you did with the B cells, right? The same idea. You had clonal expansion with the B cells. You also have now clonal expansion with the T cells. And so once this macrophage presents this, then you start to make the correct T cell. Because remember, one of the nice things about antigen presentation is that helps you to be able to identify who fits to this particular antigen, because T cells have to find the correct fit. So it's kind of like if you were to take the enemy's head back to the camp, but only one soldier has the correct training to defeat that enemy. So what you do is you basically show the antigen to all the soldiers, and you try to match up with that one soldier who has the correct skill set to beat the enemy. Once you find them, then you say, listen, whatever you have, we want to make sure we train everybody else to have it. So now we want to clone you and create an army with your skill set to go after this particular enemy. Okay. And that's basically how your immune system works. So it is very, very tactical. It's kind of a little weird how close to military your immune system is. And we all throughout military history. Matter of fact, I'm actually reading about a military history right now. When the Egyptians were doing battle with the Hyksos, when they expelled them, who were largely in the Egyptian Delta. So it was basically you had to attack them with Navy, because you had waterways. They were surrounded by water. You couldn't defeat them with a land force. So that's a perfect example of finding the correct skill set. Now that probably made no sense to any of you guys, except for those of you who've actually read into Egyptian history. But maybe it'll stimulate some of you guys to look up what a Hyksos is and to look up some of those famous battles. Okay. So basically what's going to happen then is your T cell receptor is going to bind to that specific antigen. It's going to activate the T cell. It's going to go through clonal expansion, just like we saw with the B cell lineage. In this case, it's not going to create plasma cells and memory cells. It will create memory cells, but instead of creating plasma cells, it's going to basically create what's called cytotoxic T cells. So these are the guys who do the killing. So that's basically the ones who will do that work. And so are the ones who are doing the hand-to-hand combat. That's what these guys are doing, the cytotoxic T cells. Okay, so these are the active warriors with the sword in hand. Okay, and then also some of them will create your helper T cells, right? These are the ones who are regulating and controlling it. So these are basically the tactical officers. So this is kind of what it looks like. So here you can see you've got a whole host of T cell population, each with their own specific, unique T cell receptor designed to fit to their own specific and unique receptor. Your macrophage is going to gobble up the bad guy, going to break up those proteins and then display them on that MHC1 complex, right? That's your antigen presenting complex. The other one is for your self tag, right? That's the HLA2, that's the HLA1 rather, and A, B, and D are. And then what's going to happen is it's going to locate the correct fit between the T cell and the antigen. So it's going to lock onto that. When it does that, it's going to activate it just the same way we saw with the B cell lineage. Then from there, you're going to go to clonal expansion, which is basically going through cell division, in which case you're going to be creating tons and tons of cytotoxic T cells, which will go and do their battle. And then they'll be producing memory cells that will basically allow you to retain the memory. So in both branches, both cell-mediated and antibody-mediated, you're producing memory cells, both for the T cell approach and for the B cell approach. Okay. Now, once your infection is over, then just like with B cells, you tell your T cells, listen, we don't need you anymore. The infection is gone. So go ahead and self-destruct. Okay. So that's basically what we get there. So essentially, inside the cytotoxic T cells, you have storage vacuoles, and they will contain several different proteins. One is what's called perforins, which are basically, they do what you think they sound like they do. They perforate things, right? They poke holes. In your bacteria. They also have enzymes, granzymes, which basically hydrolyze the bacteria. They digest it. That's a fancy word for digest it. So they'll do this to any cell they run into. So for instance, if you have a virus infected cell or a cancer cell, it'll release perforins. It'll punch holes in the plasma membrane, basically popping the balloon, right? That's basically what's happening with those. And then what's going to happen is the granzymes will move into the pore and they will essentially cause apoptosis. This all moves pretty quickly, right? So the pores basically diminish the structural integrity of the membrane, popping, right? Plus the granzymes will essentially cause the cell to sort of essentially break up, for lack of a better term. And so this is basically your cell-mediated immunity. So it's kind of what it looks like. So here's your attack system. So you can see a vesicle with your perforins will open up. It'll go through exocytosis, and it'll basically start to insert those perforins into the membrane, and they'll start to form this little pore-like structure. Then you can basically, first of all, you're creating an unregulated flow of fluid into and out of the cell, which will cause it to pop it, very similar to the way we saw with complement, right? Creating holes that basically dysregulated that osmotic water flow and caused the cell to pop. But now you can basically put the granzymes in there, which will then stimulate the cell to go through apoptosis and essentially disintegrate. This is what it looks like. So here's a target cell. This whole guy right here is a target cell. So this is one that's going to basically be subject for destruction. And here you can see these cytotoxic T cells, these big purple circles. These are cytotoxic T cells that are actually latched onto the cell. They're creating perforins. You don't see that piece because they're underneath. And they're basically injecting their granzyme into this cell, which will basically cause it to essentially dissipate and break up and go through apoptosis. So then, we've talked a little bit about the helper T cells, but there is still a lot more to talk about the helper T cells. One of the important things with the helper T cells is the helper T cells will secrete cytokines, and this will basically regulate the lymphocyte response. So it'll help to enhance the response of all these immune cells. B cells, for instance, will not turn on without a T cell regulating it. So you won't get an antibody-mediated response without your T cells guiding that process. This is one of the reasons why HIV, which is the virus behind AIDS, is so dangerous, because unlike most viruses, which infect other tissues or other cells in the body, HIV goes after helper T cells. It infects the helper T cells. The regulators, the tactical officers of the military. The idea being, if you knock off the tactical officer of the military, the infantry will not know what to do, and they'll be easy to beat because they won't be regulated or organized, or they won't have anybody supervising them. This is something actually that was well known in ancient warfare. As a matter of fact, it was so effective that it was considered foul play, if there is such a thing in war. It was considered foul play if you took out an officer. The idea being that you're taking out the tactical person on the field, and you're leaving the sheep to sort of scatter wildly, and they're going to be easy to pick off. As a matter of fact, we ran into this in the Revolution as one of the big complaints of the British Army as they were fighting us and having no problem whatsoever killing us. But we fought a different style of war. We didn't just line up in bright red coats with a big target on our chest and say, here, can you shoot us from that far? We were a little more guerrilla warfare. So we were hiding behind trees and jumping out from ambushes. And one of the things we did was we shot their officers. And they didn't like that, because they felt like that was, it would be, for them, it was like the equivalent of what we would consider today like a war crime. Like, there's just certain things you can't do. It'd be considered a war crime, which is if you lose, you're probably going to get tried, and you're going to get convicted, and you're going to get executed for that one. But that's kind of what the reason being is, because you can't shoot our officers, because then you're going to be removing our tactical brain trust on the field, and our soldiers aren't going to know what to do if you remove our tacticians from the field. And so the same thing is true for HIV. It fights dirty. It goes for our officers. It goes for our commanding generals. It doesn't shoot for the infantry. It doesn't waste time on the infantry. It goes for the generals. It ignores the infantry and looks specifically for the ones with the blueprints and the maps laid out, and they start shooting and going after those tactical minds. That's exactly what the helper T cells are. Okay. And that's why HIV is such a dangerous virus, more so than most. So T cells, then, some basic kind of wrap-up characteristics that we have. Obviously, we have cell-mediated, which will go against both virus and cancer cells. They are produced in the bone marrow, but they're matured in the thymus, which is why they're called T cells. And this maturation process happens early on when you're little. And so by the time you're an adult, that thymus becomes fatty tissue on top of your heart. The cytotoxic T cells will basically go after anything that's non-self or anything that is also abnormal. And the helper T cells are the ones who basically secrete the cytokines to control both the T cell and the B cell response. So you notice a strategy here. The communication strategy is associated with these little protein-like signaling molecules called cytokines that regulates and kind of controls all of these cellular responses. Very much like a tactical officer. These communications need to be very, very robust in order to communicate changes in strategy on the field. By the way, you actually see an evidence of this strategy. How many of you guys have seen Saving Private Ryan? Nobody. Or nobody wants to admit it. If you want to see a movie that'll show you exactly what war is like in all of its hideousness, no punches pulled, right? It's like one of those things where it's like, you need to be warned before you watch it, because it is about as close to what the real soldiers saw on that beach as the cinema can ever get. It is to World War II, at least D-Day, what Schindler's List was to the Holocaust. Okay, so it is a no punches pulled, brutal peel back of what exactly it was like. One of the things, the characters on there was a radio man. And he's one of the ones that made it off of the beach. And a lot of times people would be like, well, why does it have a radio man there, right? I mean, it's like, this is like a communication. He just ran around with like a little radio, and he wasn't like one of the other combatants who were like, all tricked out with their packs and their helmets and their guns. And obviously they were fighters, they were infantry, they were the ones shooting and doing most of the grunt work. This guy was running around with a radio in his hand. And a lot of times people are like, what's the radio guy? I mean, why did you have this guy? And it's like a pigeon. He's just like sitting there, sit and duck. I mean, you're just asking for this guy to get picked off. And the reason is because he was the tactical guy. So the reason why you have a radio man in there isn't because you want to listen to music. The radio man is essentially the eyes and the ears of the battalions and the companies that are storming. So the soldiers that are fighting are getting their instructions from the radio man. The radio man is the guy who is hearing from the tactical officers who are typically away from the battle and safe, and relaying instructions, real-time instructions, into the radio man, and the radio man is supposed to be relaying those to the commanding officer. The problem with D-Day is most of those commanding officers were picked off the beach, just like a first-class private, right? So a lot of times you were losing your commanding structure. In this case, you still had your radio man there. That's what he was there for. He was part of that tactical piece, so the tacticians could relay new instructions into the battle so that the soldiers knew what to do. Okay. Now we can move into an area of immunity, acquired immunity, typically. Now, there is what's called passive immunity as well, and active immunity. These are two different types of immunity. And so when you take a look at this, you can either work to get it, or you can basically have it gifted. So those are the two types of immunity. Most of the immunity, for the most part, is acquired, right? So what you have in your immune system and your profile of antibodies is something that you have essentially developed over your life, and you've earned those stripes, right? So it is, your immune system is a record of all the things that you've encountered in your life. And so it's all acquired in nature. It's like an experientially acquired system. So active immunity basically is when you cause the body's immune system to make its own antibodies, to basically mount an immune response against a particular antigen. For instance, this either can happen through normal infection, like you get the flu, for instance, and you build antibodies toward that when you beat it up, or you can get it through vaccinations. And this is kind of what we had with COVID, right? So we had early on, before we had vaccines for COVID, we had individuals who were getting COVID, surviving it, and then they had antibodies against COVID. And for a while there, if you recall, there was some talk about how people could donate plasma, their antibodies, to help people keep from getting infected. And so that would be a type of kind of a natural infection. And then once they develop the vaccine, then that would be a type of artificial infection, or artificial infection, quote unquote. A vaccine isn't really an infection. It's more of like it's more like a war game. A natural infection is like an actual battle. There's a real enemy. Your borders are breached, so to speak, right? They've invaded your territory, and you have to mount an attack and a response, right? That's what a natural infection is like. You're actually beating back the enemy. A vaccination is a little bit different. A vaccination isn't like the enemy coming into your territory. A vaccination is more like war games. So this is basically where you have kind of like a mock invasion that's robust enough to where you're able to train the different pieces of your immune system to know how to respond should a scenario like this occur. Okay, that's kind of what a vaccination is like. So ultimately, in either case, you're going to have antigens that invade your body, either from the pathogen, or from the vaccine, if you will, right? And so these guys um, will ultimately be what triggers your immune response, and then it forces your body to make antibodies against it. Typically speaking, when you're taking a look at exposure, and for the most part, I think we're going to talk first of all about natural infections first, and then we'll take a look at the whole vaccination cycle. It's kind of a little bit the same way. You kind of think of them the same, because with the vaccinations, what you're trying to do is you're trying to mimic a natural infection without the symptomology of a natural infection. So that's what you're trying to do. So there's a lot of similarities. Either way, whenever you get that first exposure to an antigen, it produces what's called a primary response, right? And so ultimately, when you take a look at your primary response, usually your IgMs are the first responders. Remember we mentioned that? So the IgMs are the first responders here. And then we're waiting for the IgGs to get selected out, because those typically are the ones that are the dominant ones in your blood that represent your long-term immunity, your IgGs. And then what happens is we have to basically go through a second exposure where you get a secondary response. And so ultimately, the reason why you have two responses is because you have a differential response depending on what your memory lymphocytes and some other sorts of things are like. So for instance, if you have some memory lymphocytes, then it's going to change that second response nature. Or sometimes if that primary response does not give you a strong enough immunity, then oftentimes you'll need to expose your system to a second response to try to bolster that immunity. And that's what we refer to as a booster. So that's kind of what it looks like. So usually we'll give you your first vaccine. And then generally speaking, if you take a look at your plasma concentration of your antibody, you'll see that you'll get a little bit of a response, but not a very strong response, right? So it's a weaker response in your primary response. And then what'll happen is generally within a couple of months, that'll go back down. And then we give you a booster. Now, the booster, because you've kind of already primed the pump, your immune system already has kind of a little bit of familiarity with it. Then what happens is the booster has a much stronger response. So once you get to put the booster in there, then you're going to get a much more dramatic antibody production in the second response. And oftentimes that'll also go down, but usually it won't go down all the way. You'll maintain a particular level, what we refer to as your titer. So your blood titer, what that basically means is the amount of antibody in your bloodstream. Now, in some cases, and it depends on the reaction of your immune system, in some cases, what happens is that first response is robust and it gives you immunity like right away. This is those vaccinations that you get that only require one shot and you're good, right? And you're good like for a lifetime. I mean, like your immune system responded to it so strongly that it's like, okay, no, I got it. Yep, I got it. So I'm there. So you have that, and you're gonna have a titer for that for the rest of your life, right? It's like measles, mumps, and rubella, right? Those usually you're not supposed to have those more than once, but right. I mean, the idea is you get those when you're little, and they should last you a lifetime, right? And then there's other things where it's like, yeah, no, you kind of need a periodic booster for this one, like DPT, right? Or tetanus, diphtheria, pertussis, and tetanus. It's also diphtheria and pertussis, but tetanus certainly you have to have every 10 years, because the idea being that the response, that initial response, is good enough to kind of give you titer levels, but it kind of slowly diminishes over time until like after about 10 years, it's like, yeah, you kind of need another booster to kind of get these levels back up again, right? This is the same thing we had with COVID, right? A lot of us, when we came back, we could only come back if we had the COVID vaccination, right? And so a lot of us got that initial, but then what did we have to do after that? We had to go back a few months later, right? And we had to get the booster. And some of us got a couple of boosters. Some of us may have 3 boosters. Maybe some of us be routinely getting boosters overall. because now it's kind of dropped into the endemic phase, so it's more like a flu shot now every year. But the idea being that that initial shot basically had a first response that kind of crested, provoked your immune system to attack it, and it was giving you enough cover for a few months. But then after a few months, you needed kind of another extra kick to get that titer level up sufficiently where we felt like it was going to keep you safe for a longer period of time. And if it dwindles down again, then you get another booster, right? So you basically get as many boosters as you can to kind of boost it up and to kind of keep it at a level. Now, some antibodies will kind of dissipate over time, and some you'll just hang on to for life. They're like the hepatitis shots. Those are ones you get once and you're done, right? And so there's others that are like that. And there's some that you just constantly have to keep getting boosters. It's just the way the actual individual immune system responds to that particular antigen when you get that shot. And it's very individual. So passive immunity is going to be basically something where you are gifted your immunity. For instance, when I mentioned the COVID person who had COVID and got over it and wanted to donate plasma because that's where their antibodies are, that if I got antibodies injected into me, which some people did actually during COVID before Pfizer came out with their vaccine, they were getting antibody injections from people who had had COVID and survived, and they developed antibodies against it. That would be a type of passive immunity. And by the way, that's not an invention of COVID. We had antibody vaccinations before then, just that they weren't as prevalent, because oftentimes we usually prefer to give you an active immunity. We want your body to store the memory of this, to actually go through the war games of this so that they can remember how to fight the battle when it shows up. And that's the reason why we tend not to do passive immunity, because you don't really learn anything from passive immunity. It's kind of like me giving you a test and giving you the answers before I give you the test. Like, I'll pass out the answer key to you with the test. So what I did was I just gifted you the solution to the problem. But did you learn anything? No, you didn't learn anything, right? But if I make you study for the test, then the idea is you learn something, and then when you see a particular question on the test later, then you realize, oh, okay, I learned that one because I had to learn that one beforehand, right? That's the whole idea behind these Connect assignments, right? These SMART assignments on Connect. The idea is you kind of wrestle with those questions in that sort of developmental space, so that when you see it on the exam, you're like, oh, I recognize this question, got the answer to that, right? So you're not suffering as much on the exam, hopefully. Then you're doing it all under the guise of a vaccination. So basically, anytime you're given antibodies, that's a passive immunity. For instance, newborns are given their IgGs, right? Those are crossing the placenta. That's a gift from mom to the baby. So those IgGs are coming into the newborn. And then not only that, but remember, you have IgAs, which are also coming in. And those are also being gifted to the baby because the mother is giving that to the baby through the breast milk. And so those are all gifts. Anytime you get it without doing anything for it, that's basically a type of passive immunity. So here you can see the IgGs passing from mom's blood, going through the placenta and into the baby, giving the baby her immune coverage. So whatever mom's immune to, the baby is immune to, as long as the baby is inside the mom. Now then, adding to that, you're delivering both IgGs, because mom's also making IgGs, right? That's also in her breast milk, but also IgAs, which are also in breast milk. Those are being secreted and into the breast milk and being delivered to the baby. And the baby's digestive system is not fully developed when they're breastfeeding. This is one of the reasons why antibodies can get in there and kind of help protect the baby, because oftentimes, remember we said that breast milk is exposed to outside pathogens potentially, and so this kind of gives that baby some coverage so that they're not quite so at risk. And then of course you can have that delivered directly by injection or vaccination if you get an antibody injection. And so your cytokines basically kind of pull it all together. These are the ones that are essentially regulating all of your T cell lymphocytes, your macrophages, and other cells. These are also some of the molecules that are being produced by those cytoplasmic granules. Of your granulocytes. So they regulate not just your T cells and your B cells, but they also regulate your other white blood cells. And they'll regulate a lot of the responses, like for instance, interferon responses of an infected viral cell and interleukins, which are cytokines, which are basically communications or communication molecules between white blood cells. So as the white blood cells are going back and forth, talking to each other and coordinating their attack, whether it's on a non-specific or on a specific type of an attack, whether it's adaptive or not, then these molecules are kind of pulling them all together. So you see this communication strategy with interleukins and cytokines basically pulling and coordinating all of these, whether you're in a specific type of attack, an adaptive immune system, or a non-specific attack. They're all communicated and pulled together by these cytokines, which is coordinating all of these attacks. Okay, so this is a good place to stop. So what we'll do is we'll begin talking about some disorders of the immune system. Of course, one of the first places we're going to start is HIV.